Good morning. My name is Jana Hexter. I'm with the Northeastern IPM Center based in Cornell University in Ithaca, New York. It's actually a beautiful day here. We have kind of spring-like weather today, so it's a wonderful respite from the cold weather for a couple of days. So we have a wonderful agenda for you this morning with some great presentations. And if you haven't participated in this before, it's kind of a fun and fast-paced. So the format is that we have 5-minute video presentations. We have two, and then we have a 5-minute break for questions from you for our presenters. Then we do two more. We have a 5-minute break for questions. So it's meant to be very, very interactive. And so to facilitate that, I ask that you put your questions in the Q&A. Helpful for me if you can also say who your question is for, since we have 12 presenters today. And then when we get the Q&A breaks, I can field those questions. Feel free to chat amongst yourselves in the chat. I won't be monitoring that. I'll be paying attention to the Q&A. As you saw in the slide that I just flicked by, there is going to be a recording of this, and anyone who is registered will receive a recording. And with that, I believe, yes, we'll go back and I'll stop the share so we can see the very first presentation, which is by Flor Acevedo. And it's an update on the development of action thresholds for managing spotted lanternfly in vineyards.
Hi, everyone. I am Flor Acevedo, an assistant professor of entomology at Penn State University. And I will provide an update on the estimation of economic thresholds for spotted lanternfly in grape. This is a project that we have been conducting for the last 3 years. And in this first slide, I would like to acknowledge the team working on this project. Spotted lanternfly is an invasive species that has become a new pest in vineyards in the US. This insect affects fruit quality and also plant health. Therefore, it must be managed to avoid crop loss. All insect stages, eggs, nymphs, and adults can be found in vineyards, but the adults are the ones that are more problematic because they are more mobile and they extract large amounts of plant sap during feeding. So we know that these insects have to be controlled, but we don't know the number of individuals that will cause economic damage in grape. So the objectives of this project are to first determine the loss in yield and juice quality caused by various spotted lanternfly densities, and second, to determine the number of lanternflies that should trigger management actions. To achieve these objectives, we use 20-year-old Cabernet Franc vines. We built enclosures to contain the plants and the insects, and we infested these plants with different insect densities. The densities that we use range from 0 to 15 individuals per shoot, and these vines have been reinfested with the same number of insects for 3 consecutive seasons. These are our treatments. Some grapevines were infested with only nymphs early in the growing season, and some with only adults later in the season. And also we had control plants without insects, some with cage and some without. And these are our results. We found no effect of either nymphs or adults on yield during the first year of infestation. During the second year, we found a decrease in yield with the adult infestation. And in the third year, the yield decreased even more with the adult treatment. When we look at the effect of spotted lanternfly feeding on juice chemistry, the first thing we noticed was a decrease in fruit color for this red cultivar, where fruit and juice coming from insect-infested plants were more pale, than the juice from control plants and from plants exposed to lower insect densities. So we measure different juice chemistry parameters, including pH, sugars, phenolics, and tannins. And this is a summary of what we have found so far. We found a decrease in juice pH and sugar content for the adult treatment during the first and second year of infestation. We also found a decrease in phenolics and tannins for the adult treatment. And for nymphs, we found a decrease in sugars the first year of infestation. The samples from the third year have not been analyzed yet. And based on our results, we draw these preliminary conclusions. Spotted lanternfly infestation decreases grapevine yield and wine quality. And in our experiments, there was a reduction of sugars, phenolics, and tannins in grape juice after a single season of spotted lanternfly infestation. There was a decrease in yield in the second and third year of infestation. And it looks like adult infestations appear to be more damaging than those from nymphs at the densities that we use in our experiments. Lastly, I would like to acknowledge the funding agencies that include the College of Ag, the Penn State College of Ag, USDA, the Pennsylvania Department of Agriculture, and the Pennsylvania Wine Marketing and Research Board. And I would also like to thank the Northeast IPM Center for giving me this opportunity to share my results. Thank you so much. And if you have questions, feel free to email me. My email is in the first slide. Thank you.
Thank you very much. And Flora is actually with us, so she can answer your questions if you have any after the next presentation. And the next presentation is by Cameron Cedeno. He's from Pennsylvania State University, University Park. And the title of his presentation is Resistance Among Arugula Cultivars in Pseudomonas cannabina. Alisa Lenis, and well, I'll let him pronounce all of that as he goes through the presentation. There we go.
Hi, my name is Cameron Cedeno, and today I'll be providing a quick update from the first year of the Northeast Arugula team's research. Before I get into it, I just want to say thank you to Northeast SARE for providing funding for our research project. So, the Northeast Arugula team, acronym NEAT, we call ourselves the NEAT team. In very short terms, our goal is to improve profitability for arugula and brassica leafy green growers throughout the Northeast. We aim to do this through a combination of research and education. There's me there, and this is the rest of our team. So, our research goals are listed here. I will go through these very briefly. One of our main goals is to evaluate commercially available arugula cultivars for disease resistance to bacterial blight and black rot, both significant diseases of arugula and other brassica leafy greens. As well, we aim to do disease sampling for any growers who are interested, so we can either figure out how to set up us going out to their farm to scout and sample disease, or we're more than happy to help growers send in samples for us to evaluate. As well, growers are also interested in if there are any arugula cultivars that are resistant to flea beetles, one of the more significant foliar pests. And as well, growers are interested in which arugula cultivars bolt faster than others. So these are all things we aim to address throughout our research. So a little bit of arugula background very quickly. There are 3 significant species of arugula that you can find on the market. Probably your most popular here on the left is the salad arugula. In the middle here, you find your wild arugula. That's typically considered spicier and with more of a mustardy taste. And then on the right here, this is a relatively new type of arugula to the market, and it's called wasabi arugula. Down here, you can see the breakdown on how much of each of these species we included in our research. So here are the results from our summer trial earlier this year. We did do a repeated fall trial, but that just wrapped up last week and the data is not quite ready yet. Very quickly, just going through this on the top here, we have the results from the bacterial blight plot, and on the bottom we have the results from the black rot plot. In very short terms, we are seeing more disease in the salad arugula varieties versus the wild arugula varieties. Then going on to our greenhouse inoculations. So we try to mirror the same things we do in the field as we do in the greenhouse. On the top here, this is the results from our summer field inoculations with the bacterial blight pathogen. On the bottom here, we have the greenhouse results from the same kind of experiments. So we also did greenhouse experiments with the black rot pathogen, but again, those also just wrapped up last week and the data is not quite ready. But as you can see, the results are relatively mirrored in the greenhouse as they are in the field. We see a bit more varying results in the greenhouse for the salad arugulas, but overall they still seem to be heavily susceptible compared to the wild arugulas. So for this video, we were asked to talk about one thing that we think is really cool, and this is it for me. So a thing I love about our research is that we get to include other Penn State branch campuses. So way back when I was a student who transferred from Penn State Beaver to Penn State, Maine at University Park to finish my degree. And now I'm in a position where I can work back with the same student farm that I used to work with. And we send them arugula seeds, same with the student farm at Penn State Brandywine and at Penn State, Maine. They grow up arugula, and then if they get any disease, they let us know about it, and then we coordinate them sending samples up to us. It's something I really love, and I think it's really cool, and would love to talk more about if anyone asks me. So that was a very quick update from the Northeast Arugula team. I just wanted to take this opportunity to share my contact information in the top right corner. In the top left, we have a QR code that will send you directly to our website. Below that, we have a QR code that will send you to our arugula production survey. And I want to take this opportunity to ask everyone to please share this with as many people as you know who may be either directly involved with arugula growers or who could help connect us with arugula growers. We're asking growers to take that survey there, and then we have the link to our website for more information. Thank you so much.
Great, thank you very much, Cameron. Excuse me. So if you have any questions for Flor or Cameron about their presentations, please put it in the Q&A. I actually have a question for Flor, actually. Flor, you showed that graph where there was an effect, there was a lowering in year 1, but no effect in year 2. Does that mean it was lowered in year 1 and continued to be lowered in year 2, or it was lowered in year 1 and then it was back to the normal level? There was a couple of them on your chart that showed that.
Do you mean for yield?
Yes, I think it was the yield slide.
Yeah, so it was not significantly different for the first year. So we only found a significant effect on the densities, on the lanternfly densities on yield in year 2 and above.
Mm-hmm. But for there was a few, and I forgot what the measure was, but you were pointing where things were lower in year 1, but then no effect in year 2. And so I'm just wondering if that meant it was just flatlined from the lowered effect in year 1. And I should go back and look at the slide.
I should be looking at the slides as well. It was not significantly different. I don't remember if it was flat or if it was still low, but just not significantly different. I will have to look at the slides.
Okay, all right, thank you. And Surinder Chopra said, is there any HR or HR-like response at the feeding sites of nymphs or adults?
We have not tested that, but that's interesting.
Okay.
Yeah.
Great, thank you. All right, well, I don't see any more questions. We have lots of presentations, so we're right on time to dive into the next one. And that is by Veronica Yurchak from the University of Maryland. And she's presenting on creating an eco-friendly pest suppression program in sweet corn funded by Northeast SARE.
Hi everyone, I'm Veronica Yurchak, and today I'm going to be telling you a little bit about one aspect of our project titled creating an eco-friendly pest suppression program in sweet corn. So this project was very broad. And so first we were looking at using different cover cropping tactics for weed suppression in sweet corn. Then we wanted to measure how this change in plant diversity that resulted from these different cover cropping tactics influenced the insect natural enemies as well as pest damage. And then finally, how one of our treatments in particular, which contained a flowering living mulch, influenced the pollinator community within that sweet corn field. And so today I'm just going to share a few of our findings related to this sort of pollinator focused aspect of the project. And so for this objective, we wanted to see if the presence of this flowering red clover living mulch between our sweet corn rows, which you can see in the image on the left, if this would increase the abundance of bees and butterflies compared to more conventional bare ground production, where you can see in the image on the right, there really are very few food resources for pollinators during most of the growing season. And so to do this, we performed visual observations of all bees and butterflies feeding on the red clover flowers throughout the sweet corn growing season, as well as observations of pollinators collecting sweet corn pollen during that pollen shed period. We also used bee bowls or pan traps to measure the species richness and diversity between our different treatments. And today I'm going to focus on the results of our visual observations. So for the visual observations, we performed these observations for 5-minute periods, once in the morning and then again in the afternoon on a weekly basis in the red clover, and then on 3 separate occasions during the sweet corn pollen shed period, where we recorded what we observed visiting those sweet corn tassels. And so I want to quickly mention that many wild bees are very difficult to identify in the field. And so for those bees, we grouped them into morphospecies categories. So when we look at our results, that's why we see things like bumblebees or honeybees for those that are easy to identify, but also categories like small dark bee or metallic bee. And so we use those bee bowls or those pan traps that I mentioned to better understand what species were out there that we were categorizing in these different groups. And so if we look at this graph, this shows the average number of observations that we made within that 5-minute period over the entire season. And so what we saw was that it was primarily bumblebees utilizing those red clover flowers. So we saw a lot of bumblebees visiting the red clover, followed by skipper butterflies, and then also quite a few honeybees, white and sulfur butterflies, and brushfoot butterflies. And then finally, a less common but still very prevalent assortment of other wild bees and butterflies, including monarchs and swallowtail butterflies. And so quite a diverse community of pollinators feeding on those red clover flowers. Looking at the sweet corn tassels, first I want to mention that we didn't see any treatment differences for what we observed visiting the tassels. So we performed these observations in all of our cover crop treatments as well as the bare ground, and we found that it really didn't seem to matter whether we had that additional flowering living mulch lower down in the canopy when it came to what was collecting that sweet corn pollen. And so for that, we found that it was mainly honeybees collecting the sweet corn pollen, and then a handful of observations of large dark bees, which we later determined to be the 2-spotted longhorn bee, as well as some pollen collection going on by our different metallic or halictid bees. But overall, we found that interplanted red clover provides a food resource for numerous bee and butterfly species, which otherwise were not found in those sweet corn fields. And so it was generally a very different, more diverse community than what was found in the sweet corn tassels during that pollen shed period. And so with that, I just want to say thank you, and that I'd be happy to answer any questions that anyone might have.
Lovely. Thank you very much. And our next presentation is by Leo Kerner from the University of Maryland as well. on using red clover to recruit natural enemies to control pests in cantaloupe.
All right. Hello, everybody. My name is Leo Kerner. I'm a student of Dr. Hooks in UMD's Department of Entomology, and today I'll be talking about recruiting natural enemies to control cucumber beetles using a living mulch. So striped and spotted cucumber beetles are both pest species of cucurbits. Today we'll be talking about cantaloupe in particular. So these beetles damage fruit, roots, leaves, and flowers of cantaloupe, which can reduce productivity and marketability of fruit. More importantly, though, both species transmit bacterial wilt through their feces and through feeding, which is a bacterial disease that can have particularly high mortality in cantaloupe, so reducing beetle interactions with plants is vital for cantaloupe growers. Our objectives for the study were to reduce cucumber beetle presence, to increase natural enemy presence, and to increase rates of parasitism. And our goal was to do this through the use of a living mulch. So a living mulch is a cover crop interplanted with a cash crop remaining as a living cover for the duration of the growing season. And usually this strategy is for weed suppression, but for the sake of our project, we were interested in how a living mulch could improve habitat modification for natural enemies. So our system is cantaloupe that is grown alongside a red clover living mulch. We figured this would be a good combination because cantaloupe has a long shading vine that might be able to grow over top of the low dense growth of red clover. So reducing competition between these two plants while still maintaining a suitable habitat for natural enemies via the red clover. Our methods were a complete split block design in which we used plastic mulch as our conventional cantaloupe growing setup as our control, and our other whole plot was our red clover living mulch. Each one of these whole plots was divided in half, where one half received an insecticide treatment, which was imidacloprid, fed through irrigation, applied once in the growing season, and the other half received no imidacloprid. Our hypothesis for the study was that the living mulch whole plots would provide a more suitable habitat for natural enemies and help reduce cucumber beetle presence. We tested this hypothesis by collecting beetles and natural enemies with pitfall traps, sticky cards, counting them visually using a quadrat, and with vacuum samples. For the sake of this project, though, we'll just be going over our visual count data. We calculated rates of parasitism by collecting as many beetles as possible and rearing them for 20 days, waiting for parasitoid flies to emerge, which looks a little something like this. Now to go over some results, we found that living mulch didn't seem to have an impact on the numbers of spotted cucumber beetles, which are represented with these black dots. The plastic mulch little roll represents our conventional treatment. Clover represents our clover treatment. Each one of these, remember, was split into an insecticide and non-insecticide subplot treatment, where mean abundance is on the y-axis per meter squared. But we did find that striped cucumber beetles were more abundant in our plasticulture plots, and this was true for 2023. The pattern is somewhat similar in 2024. We also found that living mulch effectively increased natural enemies. So you can see our natural enemy abundances are higher in our living mulch plots for both years. We also found there was hardly any parasitism of spotted cucumber beetles across both years of our study. But for our striped cucumber beetles, we actually found that plots that received an insecticide treatment had a higher rate of parasitism just for these striped cucumber beetles. But this wasn't true in 2024. So some takeaways: we found living mulch effectively reduces populations of striped cucumber beetles. My thoughts here are that the red clover is maybe hiding the food source, which is the cucurbits you can see in this top picture. And maybe the greater presence of natural enemies is reducing the tenure time of these cucumber beetles. We also found living mulch effectively increased natural enemies. Perhaps the habitat in living mulch environments is more suitable for them, and maybe there's a greater diversity of prey options for generalist natural enemies. Finally, we found standard chemical treatments improve rates of parasitism by striped cucumber beetle tachinid flies. However, this result was not consistent across both years of our study, so for now we'll say this is inconclusive and more work is needed to be done. So with that, I'd like to thank many people for participating in this project and for my funding sources, and I'll take any questions. Thank you.
Wonderful. Thank you very much. So there is a question here from Bob Littletree. He said he's the chair of the Southeast chapter of the UVM Master Gardener program in Vermont. For small vegetable gardeners, excuse me, for small vegetable gardeners, would it be a good idea to surround these gardens with clover? And I will let either Veronica or Leo respond to that.
So I can give some input. So I would say for smaller gardeners, if they have the space and the resources to do it, we've shown it does seem to help boost populations of natural enemies, and it does provide a food resource for pollinators. So if you can, there's not really a downside to it. It's sort of not fully fleshed out, how consistent, the natural enemy movement into the crop is and stuff like that when you surround a garden like that. But if you can, there's generally some benefits that go along with it. I don't know if Leo has anything he wants to add there.
Yeah, I agree. I would agree in saying that, yeah, based on I think both of our studies, there seems to be benefits to planting clover. So yeah, if you have the resources, I'd say go for it. But thanks.
Great. Thank you very much. And thank you for the question, and keep them coming. Since there are no other questions, we're going to go ahead. We actually do have one presentation that didn't quite make the roster. And so if we have a little extra time, we will try and slide that in at the end. So the next presentation is by Carlos Quesada, and he's with West Virginia University. And this is reporting on a project that's funded by the Northeast IPM Center on evaluation of insect exclusion screens on pests and biocontrol agents in commercial high tunnels.
Hello, my name is Carlos, and today I will talk about effects of exclusion screen on pests and beneficial insects in high tunnels. So high tunnels are structures used to improve the growing conditions during early spring and late fall. So unlike greenhouses, a high tunnel have this opening here to control temperatures. So it has been shown that by using screens on those openings, we can increase the abundance of pests such as aphids. However, we can reduce that abundance of other larger pests such as cucumber beetle. But what about beneficial insects? My objective was to evaluate the impact of physical control on parasitoids and predators. To do that, I work with 5 farmers. Each of those farmers have 2 high tunnels, and I ask them to whatever they do in high tunnel A, repeated on high tunnel B. And then I place screens in one of the high tunnels. All the farmers plant tomatoes. Some of them plant tomatoes in combination with pepper and or cucumbers. So the weights I calculate the aphids was by placing sticky cards, counting leaves directly, or using vacuum. All the data that I will show today is my vacuum data. All my graphics are like this. In the bottom, I'll have the year, and in this exit, I will have that time where I collect the insects, and in here I will have that insect. In this case, aphids. In other cases, will be predators or parasitoids or natural enemies in general. So the lines with the black dots represent that insects in that high tunnel with the screen, and the dots on white dots represent that insects on high tunnels with our screens. So here in 2023, we can see that there was not a statistical difference between that high tunnel with our screens. However, in 2024, we can observe over here that high tunnel with screen increase the number of aphids. So this is consistent with the data that have been already published. Now let's see what happened with the natural enemies. So the natural enemies were significantly reduced by the screens. And this is for 2023, and we have the same result consistently in 2024. Now, I was interested to see what happened with parasitoid wasps, because they're very small and they can go through the screen. So we found that screens reduce the parasitoid wasp in both years consistently in 2023 and also in 2024. And we observed the same patterns for our predators. We observed the predators were lower on a high tunnel with a screen, and this also was consistent in 2022. So 2024, sorry. So we found that a screen increased abundance of aphids, which is consistent with other publications, other research, but we also found that parasitoid wasps and predators abundance were reduced by screens. So due to the effect of these screens on natural enemies, we believe that there is a potential of using augmentative biocontrol in high tunnel with screens. With this, I would like to thank the students who helped me with this and the funding sources. Thank you.
Thank you very much. And our next presentation is by Simon Zabilo. He's with the University of Maryland Eastern Shore, and he's reporting on funding from USDA NIFR EIP program. And his presentation is Empowering Small-Scaled, Limited Resource, and Other Farmers with IPM Knowledge.
Good morning. This is Simon Zabelo from the University of Maryland Eastern Shore. I'm presenting about empowering small-scale limited resource and other farmers with IPM knowledge. This is a USDA NIFA CPPM EIP project funded in 2021. Here are our team members from the University of Maryland Eastern Shore, University of Maryland Eastern Shore Extension, University of Maryland College Park, and University of Maryland Extension. This is how the EIP project is distributed: IPM implementation in specialty crops, agronomic crops, and animal agriculture. So these are a long list of the outcomes of this project. More outcomes of this project again, but I will focus on ethnic crops. Delmarva Peninsula has a highly diverse demographic. There is strong demand for ethnic crops. Ethnic crops are crops that are not traditionally grown in the US but imported to the US from different countries. currently grown on a small scale to satisfy the need of various ethnic groups in the US. The increased demand for locally grown foods provide opportunity for small farmers to grow ethnic crops to serve the growing diverse population on the Delmarva Peninsula. Here are the current list of ethnic crops grown in the US, including the Delmarva Peninsula, but I will focus on Amaranthus, or African or Jamaican Kalalu, Amaranthus viridis, also known as Kalalu in many countries. These Amaranthus viridis are different from Amaranthus palmer or pigweedus because they belong to different species. This ethnic crops is mainly cultivated in Asia, Africa, Latin America, and the Caribbean. They are drought tolerant and does not require a lot of inputs to produce a quality crop. They are highly nutritious and have pharmacological properties. Pigweed flea beetle is the #1 insect pest of vegetable Amaranthus. As you can tell from its name, pigweed flea beetle also a biological control of a pigweed in many agricultural systems. Interestingly, this insect is very damaging for small farmers who grow this vegetable Amaranthus. As far as my knowledge concerned, no pesticide registered in Maryland to control this insect because it's been known as a biological control for a long period of time, I guess. So here we are testing some organic and conventional insecticide if they offer some control. And also eventually we will work with IR4 project to try to register some insecticide which will help the small farmers here in Delmarva region. So we tested neem oil, spinosad, and 7. As you can see, the total number of these insects significantly reduced by spinosad and 7. And the average total yield, there was no significant difference. However, the average non-marketable yield was very low when we use 7. So we use hemp cannabis sativa as a trap crop. So this trap crop was planted at different distance point from the Amaranthus, that is 3 feet, 6 feet, and 9 feet from the Amaranthus plants. The average weekly number of insects per plant per plot was lower on the trap crop plots, as you can see, but higher in the control plots. The average weekly yield of the amaranth plant were higher on the plots with hemp than the control, particularly when the hemp plants planted 9 feet from the amaranthus crop. I am grateful to my collaborators from the University of Maryland Eastern Shore, University of Maryland Extension, University of Maryland Eastern Shore Extension, of course, in my collaborators from the University of Maryland College Park, and also I'm grateful of the funding that we received from USDA, NIFA, the CPPA IP projects. Thank you. Please send me an email if you have any questions. Thank you.
Great, thank you very much. So we have a question for Carlos, and I see that he's here. Do you have any ideas on how to increase the number of parasitic wasps inside the high tunnels while using the screens?
Yes, there are 2 ways. One is called augmentative biocontrol, which basically we buy biocontrol agents from different sources. And the other one is conservation biocontrol, which the other 2 presentations before mine were talking about planting flowering plants so they can have other sources for food. And something I didn't talk today because we didn't have enough time is that I am seeing this interesting pattern where when we use a screen, the high tunnel seems to be related to the height that the parasitoid seems to have a relationship with that aphids, and we're seeing that more aphids, the more natural enemies we're having. So that's why we think there is a potential there for augmentative biocontrol.
Okay, great. Lovely. Thank you very much. And we have a question for Simon, but I don't believe he is here right now. So maybe if he jumps on at a later point, we can move ahead there. So we will move ahead, and please put your questions, any questions that you have as we move through. We have a break every 10 minutes. So our next presenter is Charles Colvin, and he is with Penn State University, and this was funded through ARDP. And the title of the presentation is Investigating the Insecticidal Properties of Maize Flavonoids Against Fall Armyworm. There we go.
Hello, my name is Charlie Colvin. I'm a third-year undergraduate student at Penn State working in the Tropomae Genetics Lab. I'll be talking today about our project that investigates the insecticidal properties of maize flavonoids against fall armyworm. So I think we're all familiar, but in case we're not, fall armyworm is a chewing insect pest native to South and Central America. It's invasive in Africa, it's a seasonal pest in the Northeast US, and it feeds on a bunch of different crop species. The one that we work on, maize, can be highly impacted, but is typically protected by the use of Bt traits in these crops. Now, unfortunately, Bt is not suitable for organic systems, as we all know, and there's also been concerns about resistance developing among the caterpillar populations that allow them to tolerate Bt. And so because of this, there's been a big push to develop alternative ways to control this pest. And so a couple of years ago, my lab came out with a paper that showed that flavonoids produced by maize and sorghum are an effective way to control this insect. The insects that feed on a high flavonoid maize line or a diet made from these flavonoids are typically much smaller and exhibit much higher mortality than the insects that feed on a control diet. And so the mortality data would look something like this, where you have maybe 40% mortality of the insects feeding on a high flavonoid diet and somewhere around 0 for those feeding on a control diet. Now, that's great, but at the time of publishing this paper, we didn't really understand the mechanism. We just knew that the insects were dying, but we couldn't tell exactly why. And so the direction I've been taking this project is trying to isolate exactly what the mechanism is that causes these insects to die when they feed on a high flavonoid diet. Now, we believe that this mortality might be related to the microbes that exist in the insect's gut. And so to test this, we tried a feeding experiment where we fed 2 groups of caterpillars, one that had a normal gut microbiome and one that had the gut microbiome abolished by the use of antibiotics. And actually what we saw was the ones that fed on the caterpillars with the normal gut microbiome that fed on flavonoids had the same mortality that they always had, somewhere around 37%. But when we introduced the antibiotics into the diet and eliminated the gut microbes, that mortality dropped all the way down to 0. And so we saw that the presence of gut microbes is actually vital to the effectiveness of the flavonoids at killing the insects. So this led us into doing a microbiome analysis where we looked at the different bacterial taxa present in the guts of the worms after feeding on the different diets. And so all of these bars on the left here are all insects that were fed on a low flavonoid maize line, B73. And then all the bars on the right were insects fed on a higher flavonoid maize line. And so there's a couple things that jump out, but the main one here is this bright red taxa, which I've hidden the name because it's not published. This bright red taxa that's only present in high abundance in the high flavonoid group. And looking at the literature, this particular taxa is a known pathogen in several other species and some insects. And so we thought that this could potentially be the candidate that's causing the mortality and sort of a pathogenic infection of these insects. And we know by the literature that this particular bacterial species tends to target and damage epithelial tissues. And we look back at our paper from a couple of years ago, we saw that one of the things we found in the low flavonoid versus high flavonoid larva was damage to the epithelial tissue, specifically the peritrophic matrix, which is kind of the lining of the midgut. And we saw that the ones feeding on the high flavonoid diet had the high mortality, had a bunch of damage and lesions to this matrix, while the ones feeding on a low flavonoid diet really had no major damage. And so this to us kind of supports the idea that maybe it is this particular insect, or this particular microbe that's causing the mortality in the insects, because there's the characteristic damage here on this particular tissue. So in the future, we are currently working on isolating the pathogenic microbes from the guts of these insects. And we'd like to use these isolates to create gnotobiotic larvae, where we introduce just one or a few strains of bacteria into otherwise sterile larvae, And then we can use those to test the impacts of flavonoids in the presence or absence of very specific microbes and figure out exactly which ones are responsible for the mortality. Kind of tying into that, we'd also like to try and investigate the mechanism by which the flavonoid diets allow for the pathogenicity. We're not sure at this point if the flavonoids are directly interfering with the insect's immune system, or perhaps they're just decreasing the competition within the gut. We're not exactly sure, but they appear to be giving some competitive advantage to the pathogenic bacteria, and so we're trying to figure out exactly how they do that. I'd like to thank my PI, Dr. Chopra, as well as PhD students Tyler and Brian, and the former postdoc, Dr. Chatterjee, all of who have been greatly helpful in this project and guiding me along. I'd also like to say, if you want to follow us, you can follow us on LinkedIn, and I'll be here for the Q&A session, but if anyone has any questions outside of that, you can feel free to contact me at this email right here. Thank you.
Great, thank you very much. And our next presentation is by Philip Fanning, and he's with the University of Maine. And this was a project funded by ARDP, and it's classical biological control for spotted wing Drosophila in the northeastern United States.
Hello all. So my name is Phil Fannin. I'm with the University of Maine, and I'm here to share some results from progress towards classical biological control for spotted wing Drosophila in the northeastern United States. Today, I'm going to share some data on our releases in New York, Maine, and New Jersey, but also some overwintering work that was done in those 3 states, and also in North Carolina and Michigan. So spotted wing Drosophila has obviously been a devastating pest for small fruit production. Biological control has always been a goal of the research program to develop good biocontrols for that species. That began with foreign explorations for parasitoids right after spotted wing Drosophila was first discovered in the United States. Through all of those foreign explorations, 3 species were brought back and studied in quarantine here in the United States. And based on the results of those quarantine studies, a federal release permit was issued for Ganaspis camorum, formerly known as Ganaspis brasiliensis, in 2022. The 2 other species, Leptopolina and Asibara, were not host-specific enough for a release permit to be granted. Now, these 3 species accounted for about 85% of the parasitism seen in infested fruit in the home range of SWD. Ganaspis was chosen because it had the highest level of specificity. Okay, so our releases here in the Northeast, we've been doing releases in Maine, New Jersey, and New York under this project. We first established our colonies in the spring of 2022, and we've been releasing Ganaspis camorum in 2022, 2023, and then 2024. In total, the project team have conducted 48 separate releases. This was done at multiple sites across each state, and in total across the project, we have released 43,000 wasps. When it comes to recovery, we mostly still find Leptopolina japonica. So this was that species that was studied in quarantine. A permit was not issued for its release. However, we have adventive populations of it developing here in the United States. We're starting to learn more about that species. Looking at some data for infestation here in Maine, specifically in 2023, what you can see here is the average percentage parasitism in fruit collections on the y-axis, and just dates across the season that we sampled on the x-axis. Black bars indicate Ganaspis camorum, still says brasiliensis here, and Leptopolina japonica. You can see our collections were dominated by Leptopolina japonica. So this is very quickly becoming a very key species, parasitizing spotted wing Drosophila in Maine, but also across the Northeast. These results are echoed in New Jersey and New York as well. So one question we have since Ganaspis camorum got release permits is whether it'll be able to overwinter here. So our team has been working on overwintering for the past 2 years. In the first year, we paired with North Carolina and Michigan. They worked at a number of sites. We worked at a number of sites here in the Northeast. And then last year in the winter of 23-24. New York, New Jersey, and Maine continued this work, working at 3 sites each. For our work, we would take basically parasitized larvae, we would initiate diapause, and then these would be placed in the field and brought in at regular intervals. So again, just for brevity, I'm going to only show some of the results here from year 2. We had a little change in our methods between years, but here in Maine, you can see that we had in all relatively low emergence from our treatment replicates. Even in the controls, we had around 11 wasps emerging per rep. However, the key thing to take home here is you do see that emergence drops off once they're placed in the field, so days of exposure in the field on the x-axis, but we do get persistent emergence or survival, even in really low numbers, all the way out to 120 days. Looking at bringing all of our data together and thinking about how this reflects with spotted-wing Drosophila survival. So when we researched spotted-wing Drosophila survival initially, when spotted-wing Drosophila was starting to become established in the United States, we found that SWD survival was observed to be low, particularly in northern states. Ganaspis canorans survival seems similarly low. Collectively, we might see early season population growth being a little bit slower due to high mortality during the winter. Okay, so that's a quick update on our releases and our overwintering. I'd just like to acknowledge all of our grower cooperators and our funding source from the CPPM program and some additional funding that we've leveraged in time. Thank you. I'll take your questions.
Great, thank you very much. I feel like you've just skimmed the surface of what you have to present. So, so I do see a couple of questions for Simon, but I do believe he is not here at the moment. So, if you have any questions for any of our presenters so far, please let me know. And you know what I'm going to do? We have another presentation that didn't get onto the schedule, and we have time to show that actually before our next presenter is due. So if we can cue that one up, it is by Brian Panek at Penn State University, and it's development of specialty corn lines for plant and animal health, and it's funded through ARDP. And it might take a moment just to get that queued up. And there we go.
Hello, I'm Brian Panek, and today I would like to talk to you about the development of specialty corn lines for plant and animal health. First off, a little background. So my research at the Maize Genetics Lab, headed by Dr. Surinder Chopra at the Penn State University, focuses on developing specialty maize hybrid lines that display accumulations of pigmentation in various tissues. As you can see in the pictures here, we have expression in stalk tissue and husk tissue and sheath tissue, and then we have variation in expression as far as the kernels are concerned, from dark purple to this wonderful red. These pigmentations are known as flavonoids, and flavonoids have a myriad of benefits for not only plant health, but also for animal health. So we're going to start off with a little bit about the plant health or plant defenses. So previous research in our lab has shown that flavonoids from sorghum and maize lines were extremely detrimental to the survival and growth of fall armyworm. We also found that this is also true for corn earworm when they're fed the husk and silk tissue. So building off of that work, I wanted to develop hybrid lines that would do the same thing, but also yield at a commercial standard. So the way I go about this is I set up these trials, and as you can see, here is a trial here. And this one has 4 blocks, and there are 10 entries in it. And this is a completely randomized design, block design. And then throughout the season, we'll come in and we will sample tissue at various growth stages. As you can see in a couple of the pictures here, the one picture here that we come in, we take whorl tissue at the V3, V6, and VT stage, and we feed them to first instar fall armyworm larvae. We get the fall armyworm in and we hatch them and we raise them to the first instar using a soy-based diet. And we do the same thing with the corn earworm. The only difference is the corn earworm are fed husk and silk tissue and not leaf tissue. The larvae are put into these individual 1-ounce plastic cups, and the tissue is placed in there with them and changed every 3 days over a nine-day period, at which the end we calculate or we record mortality and the weight of the larvae. So that brings us to a little bit of the research, and I'm just going to show you a little bit today. So this is from a fall armyworm bioassay using the flag leaf at VT stage. And entry 1, is a non-expressive line. There's no flavonoids in it. And all of these lines except Mo17 are near isogenic from one another. So what we can see here is that entry 2 and entry 9 both showed high levels of mortality, high enough to be statistically significant, and an alpha of 0.05. And this is wonderful because this is how we actually go and evaluate some of our breeding parental lines that we're going to later use to make hybrid lines. So then here we can also see a similar bioassay performed, but this is on corn earworm larvae and first instar also. And this was after being fed silk tissue. And same thing, entry 1, entry A is our non-expressive line. And we can see that there are actually quite a few lines that showed significance when we were looking at the total body weight of the larvae after 9 days. And this is really expressive of how well the flavonoids work as an anti-feed device, because a larva after 9 days of feeding that weighs 60 milligrams is probably not going to have the energy to fully pupate and develop into a mature moth, which will later go and oviposit and, make more corn earworm or fall armyworm. So after that, I just wanted to talk a little bit about the animal health benefit side. And through some collaborations in our lab, we've seen that in broiler poultry, when fed flavonoid expressing lines compared to non-flavonoid expressing lines, broiler chickens that were induced with necrotic enteritis actually saw a less severe case with the flavonoid expressing lines. They saw a lower level of mortality, and they saw actually a higher body weight. So this is really good to the organic broiler chicken industry, as they don't have to they can't rely on antibiotics. So this can actually help increase the overall health of their flock. And we've also done similar collaborations where we have partners have taken mice and used them to develop or induce colitis in them and use our flavonoid expressing lines. And we see that the mice usually recover from the colitis, have lower levels of colitis, and are overall healthier. So I just wanted to say thank you and have a great day.
Thank you very much. All right, so I don't see any questions pop up, but feel free to put them in the Q&A. Actually, I will move on, and we'll come back to a question that I see in the next break. So the next presentation is by Patrice Sisson. She's with the University of Maine, and this was for work funded by the Northeast IPM Center. It's on identifying grassweed patches in Maine wild blueberry fields using drone aerial images for post-emergent application of herbicides.
Hello everyone, my name is Patrice Sisson and I'm a graduate research assistant at the University of Maine. I'm currently taking up my master's in plant, soil, and environmental sciences. And today I'm going to talk about my thesis named Identifying Grassweed Patches in Maine Wild Blueberry Fields using drone aerial imagery for the post-emergent application of herbicides. But before we dig deeper, here's a brief background. Maine wild blueberries is a rhizotomous woody perennial that is native to eastern Canada and Maine, USA. In Maine, these wild blueberry plants are not planted, but inhabits large fields on mountaintops and in glacial outwash plains, which formed 10,000 years ago. Now, they are considered crops and are managed by growers. Within these wild blueberries is a common pest, and that is weeds. Weeds compete with wild blueberry plants due to the competition of light, water, and nutrients. At the same time, weeds are hard to detect due to the heterogeneous nature of wild blueberry fields and the weeds that germinate amongst them. To combat these weeds, integrated weed management, which is the combination of mechanical, cultural, and chemical methods, are currently employed to manage this pest. However, with the advent of new technologies, we can now integrate unmanned aerial vehicles, or what we call drones, to establish a site-specific weed management system for Maine wild blueberries, diving into precision agriculture. Therefore, my study would have 3 objectives, and each objective will be a study. My first objective is to create a weed map, which will accurately determine where and how big the weed patches are in a wild blueberry field. My second objective is to use that weed map and integrated it into a spray drone to spot spray using post-emergent herbicides. And my last objective is to document spray drone deposition and drift in wild blueberry fields. So here are the highlights of my study. The results of my first study demonstrated that weed maps can be effectively generated using drone aerial imagery with RGB sensors, or red, green, blue sensors, and processed with a computer software called Pix4D Fields. To assess the accuracy of these maps, another computer software named ArcGIS Pro is used. We generated validation points and achieved high accuracy using image-based validation data. Let's now move on to my second study, which is to integrate that weed map into a spray drone. We conducted spot spraying trials with the spray drone at 2 sites using these weed maps. At the first site, default spray parameters from Pix4D Fields led to broadcast spraying and not spot spraying. For the second site, we adjusted the swath width to 10 feet, achieving successful spot spraying. After testing the weed map, we evaluated grass weed control and operational efficiency of drone spot spraying versus traditional boom spraying. The herbicide we use in this study is clethodim. At the second site, successful drone spot spraying showed comparable grass weed control to boom spraying, with the spray drone using less water and fewer inputs. Given the challenges with drone parameters, we then conducted a third study to determine optimal drone parameters in spot spraying. This last study tested spray droplet uniformity and drift using the following drone parameters: swath widths 10, 20, and 24 feet, and droplet sizes 220, 280, and 340 micrometers. These parameters were integrated into a spray drone filled with water. Water-sensitive cards collected were drone sprayed during the field trial, then analyzed with Dropscope, a scanning software. The results indicated that larger swath widths and droplet sizes had larger droplets, thus reducing drift potential. Further studies are encouraged to have a better understanding of optimal drone spraying.
Great, thank you very much. And we do have some questions coming up at the break. Our next presentation is by Quinn Loudy, and he is with Penn State on a project funded through ARDP. And the title of the project is Flavonoid and benzoxinoid accumulation in maize, implications for fall armyworm resistance.
How's it going, everyone? My name is Quinn Loudy. I'm a master's student in Dr. Surinder Chopra's lab, and today I'll be talking about flavonoid and benzoxazinoid accumulation in maize. So we all know climate change is affecting our ecosystems in different ways. With insect pests, it's increasing the number of generations, expanding populations to different geographic ranges, and increasing the outbreak of plant diseases. On the far right here, I have a heat map depicting population density of fall armyworm expected to increase in the next 50 years in various countries of Africa. Fall armyworm is a polyphagous species that feeds over 353 host plants across 76 families. They're native to the Americas, as you see in red up in this map, and began creating huge infestations in Africa and Asia, and they're just becoming a major issue. In corn and in the southern hemisphere, fall armyworm can begin to feed around VE to V1 stages as the seedlings begin to emerge. Up in the northern hemisphere, they begin to feed on corn around V4, V5 stages. So what we do in our lab is we study how flavonoids can be used to deter pest herbivory. Flavonoids are plant pigments that act as pollinator attractants, protect the plant cells from UV stress, oxidative stress, and act as cell growth regulators. But we focus on how flavonoids can be used to deter fall armyworm, pest herbivory, corn earworm, and yeah. So here's a study that was conducted in our lab. These are 4 different lines of maize. US and NS are low producing or not producing flavonoids in any way. UE and ZMY1, they are high producing flavonoid lines. And here we can see, so this was a V6 detached leaf feeding bioassay with fall armyworm. We can see that the body weight in the low producing flavonoid lines are much higher and the mortality is much lower. And then the high producing flavonoid lines, UE and ZMY1, we see that the body weight of the larvae is significantly lower and mortality is increasing significantly. So what I'm trying to do is locate a region of the genome responsible for the expression of flavonoid accumulation in the maize leaves at early stages of maize development, around V3, V4 stages. So I have a flavonoid line that is a high producing line. I'm going to cross it into the nested associated mapping panel. And then I take the F1 progeny phenotype for early accumulation of flavonoids, use the SNP data from those individuals and correlate it with the SNP data on maize GDP. And then I can hopefully locate QTLs that will narrow down the region for that expression. I'm also interested in benzoxazinoids, which are a secondary metabolite derived from indole, most commonly found in Poaceae. Benzoxazinoids, specifically HDM-BOA, deter feeding from insect pests, and then other benzoxazinoids reduce pathogen infection. Benzoxazinoids are constitutively expressed early in the maize plant, and then as the maize plant matures, it's more induced through jasmonic acid. And yeah. So what I want to do is I want to create some introgressed hybrids with high producer and a mutant of benzoxazinoids that doesn't produce benzoxazinoids with a high producer line of flavonoids, and then just compare with detached leaf bioassays and to determine if there's a synergism between benzoxazinoids and flavonoids, see if one's more effective against fall armyworm feeding, and then just narrow it down, and maybe breeders in the future can use the region of the genome, the QTL, use this information to produce maize lines capable of deterring pest herbivory. Thank you.
Great, thank you very much. So we have some questions around the drone, and for Patrice. They're kind of interrelated, so I'm going to read the three of them. What is the pesticide capacity of the spray drone? How often do you have to refill it? And have you conducted cost-benefit analysis for drone versus boom spraying?
Hi, yes, thank you for that question. So the capacity of the drone is around 10 liters, depends on your spray drone. And we didn't have any cost benefit analysis yet. Just tried to calculate the potential savings for the herbicides. But we did see a reduction in water that is used, and also the surfactants that are used for weed control. And what was the last question again?
The last one was, how often do you have to refill the drone?
Oh, so since it has a large capacity, we're not refilling it all over again, but the battery is the problem. So around like 10 minutes, the drone has to come back to grab another battery and then recharge it. But it's just like new batteries, and then it will fly again and continue its mission. So yeah, the gallons of water is not the problem, but the battery changing was one of the constraints in spot spray.
So maybe it's using a lot of battery because it's carrying the 10 liters.
Yeah, carrying a lot.
Can't have both, so at least yet. So
yeah.
Okay. Great. All right. Thank you very, very much. So, and I have a question here for, I believe this is for Brian Panek. If you're producing sweet corn flavonoid lines, and also if you're wondering if you're working with the corn earworm SCRI project.
No, I am not working with the cornworm SCRI project. This actually just started and is very preliminary because we saw the results with the fall armyworm. And we're not working on sweet corn just yet. Some of these flavonoids we have to watch with their solubility. So like marketability with a very water-soluble flavonoid would leave your teeth stained and maybe not be the best of products. So we're going to look into those. So first, we're going to concentrate on the animal health and the plant health, and then hopefully develop the market over time.
Right, great, lovely. Thank you very much. So, all
Thank
right,
you.
that puts us actually right on time for our last two presentations. And our next presenter is Mike Monson, and this is a totally different topic, which I'm fascinated to learn about. He's with Purdue University and Rutgers Cooperative Extension. It's funded through Northeast SER, and it's early American colonial period, circa 1680s, funerary archaeoentomology as a tool for observing a local entomofaunal baseline of historic St. Mary's City in Maryland.
Hello, friends. My name is Mike. I'm a PhD student at the Purdue University Department of Entomology, and today I'm excited to share with you some current results from my archaeoentomology project studying insects from a grave at historic St. Mary's City, Maryland. Conservation paleobiology provides ways to use fossil assemblages to understand changes in the environment and to make comparisons to modern biodiversity. Insect science applied to historic contexts can provide a richness of information through landscape reconstruction, understanding pest challenges, and season of grave deposition, among other features. Together, conservation paleobiology and paleoentomology can aid IPM by understanding historic insect conditions and their changes. This project is focusing on insects retrieved from the grave of a 1600s landowner at historic St. Mary's City, Maryland. Our research questions are: how have the insect biodiversity and functional services changed since the introduction of modern agricultural inputs? Our objective is to measure these changes, and the reason we're doing this is to learn about IPM applications of paleoentomology research. Briefly, our methods include morphological and molecular identification of the archaeological material, a modern site survey of ground-dwelling and carrion insects, and a statistical mix model, as well as an outreach program. For our current results, we have identified some archaeological insects to the species level and to the species and genus level, including grain beetles, a predatory leafhopper, and some ants. While there are thousands of tiny rove beetle remains in the coffin, none were consistent with the hairy rove beetle Creophilus maxillosus pictured on the left, which was the most abundant beetle in the modern survey. A workshop we conducted for farmers produced 8 responses to a research survey where farmers and master gardeners were more likely to say that this project is valuable for sustainable agriculture compared to non-agricultural community members. However, no one surveyed said that the project wasn't valuable. From our observations, there were likely 2 major groups of insects present in the coffin. The first were relatively surface-dwelling, while the other were soil fauna that likely invaded post-burial as long as the coffin remained a viable resource. No Creophilus maxillosus remains in the coffin is consistent with what we now know about this beetle's natural history, where its original range did not include North America. However, the beetle became naturalized through the Columbian Exchange. This highlights the question of what insect performed its function prior and which species is best to support for sustainable agricultural services. All participants in the research survey had something valuable to add. Specifically, farmers encouraged more IPM-based archaeoentomology research, and master gardeners wanted more outreach. In the future, we're going to keep working on our statistical mix model and aDNA identifications, while also still conducting outreach through workshops for farmers. In conclusion, I do think that these methods can be applied to smaller assemblages with less preservation compared to the historic St. Mary's City assemblage. And while this work has significant challenges, it is very time-consuming. It can add a lot of richness to myriad investigations. Quickly, I will say thank you to my funders, the USDA SARE and Fulbright Commission of Sweden. Thank you to my advisors, my lab mates, collaborators, and my institutions. Thank you to Jana Hexter for organizing today's event, and thank you all so much for hanging out with me and listening to my presentation today. If you have any questions, I'd love to hear from you by email. If you're interested in this project, please check out our progress reports through the QR code on the top right. And if you'd like to read more about archaeoentomology and paleoentomology in general, please consider checking out my recent publication in American Entomologist through the QR code on the bottom right. Thank you so much.
I have to smile at that quote that you had on that last slide. People didn't catch it. Maybe it's not about finding the perfect lamp, but finding the lamp inside you. There's a picture of a moth. So sorry, I just had to chuckle with that. that was a very different topic than we've ever had. Our last presenter is Karl Majewski, and he's with the University of New Hampshire Extension, and it was funded by the Northeast IPM Center. And his presentation is promoting IPM practices for improved perennial forage management in the Northeast.
Good morning, everyone. I'm Karl Majewski, and I'm a field specialist in food and agriculture with UNH Extension. I'm going to be talking about a project that involves increasing awareness of IPM practices for both farmers and non-farming stakeholders. There's been a change in patterns of land use over the years, and farms are increasingly leasing land from non-farming landowners. And much of it happens to be in pastures or hay fields. On one hand, these arrangements can provide a lot of opportunities. Farmers have access to more acreage they can harvest feed from, and landowners are able to get help maintaining open space. But there's a potential for conflict too. While both producers and landowners are usually working to be good land stewards, farms might prioritize high quality forage, while landowners might be more concerned about conservation. And as a result, there might be disagreements over harvesting schedules that might impact wildlife, or over the use of soil amendments or pesticides. Frequently, there's a lack of understanding between the two parties that means that they can't come to an agreement, and the land just becomes neglected, overgrown, or unproductive. Farmers and landowners would benefit from talking more about how to best care for the land. And this project is intended to contribute to those conversations. By increasing stakeholders' understanding of IPM as it relates to weeds, they can make better informed decisions about the best way to maintain a healthy working landscape. This project consisted of a couple of different components. First, we held 2 webinars, one targeting landowners, where we covered basic IPM concepts and discussed weed management practices, and another one targeting producers, where we were able to go into greater depth, providing recommendations for managing problem weed species within an IPM framework. We also produced a publication, Integrated Weed Management for Pastures and Hayfields, a guide for landowners, that explained why we need to manage weeds, basic IPM concepts like establishing thresholds, or the importance of using a variety of control practices, and the responsible use of pesticides. Our intention here was not to write a complete detailed weed management guide, but to provide something that could inform discussions between farms and landowners. Finally, we also included discussions about managing weeds at pasture walks and field meetings that we held over that summer. With all of that, we were able to achieve our outcome of increasing people's knowledge of weed IPM practices. We distributed approximately 200 hard copies of the publication, and it's had some 600 additional online views since we put it up. In surveys that we conducted after webinars, 88% of participants indicated that they had a better understanding of weed IPM concepts, and 83% stated that they intended to implement sustainable weed IPM practices the following season. I want to thank the Northeast IPM Center for funding this project. And I also want to acknowledge several others who were involved. The original team for this project consisted of myself and Nick Rowley, who was then a colleague of mine with UNH Extension, and UVM Extension's Laura Johnson and Kristen Williams, who was the original principal investigator. After they all left and took on other positions, Nick Warren, a researcher with the New Hampshire Ag Experiment Station, came on to help co-write the publication. I'm grateful for all of these people's visions, their ideas, and their work on this. Thank you.
Great. Thank you very, very much. And I have one update from Patrice Sisson. She said she wanted to note that the drone capacity is not 10 liters, but 100 liters, which is around 27 to 28 gallons. So
Yeah. Yeah, we have like a separate spray tank where we add it to the drone. So it's around that 100 liters. Yeah, I don't want to just want to talk
Okay.
about. Thank you.
Great. Thank you very much. All right. And we have a question for Mike Munson, but I also believe he is not here today, or he might be on a line that I don't recognize. So I'm going to share some details that you will probably find interesting. If you want to connect with any of the presenters today or people of like mind, that we have this Find a Colleague site on our website where people post who they are and what they're interested in, and you can find each other through that, and you can post a profile about yourself and your work at that place. We are also always looking for peer reviewers of our grant process. We always choose people from outside of our region, but if you are interested in being a peer reviewer for another region, please go to that link and you can fill out a Google form about your areas of interest and your areas of expertise. And it's especially useful, I think, for early career scientists, because it gives you a really good idea of how the review process works. So I encourage you to go there. And we have some questions for you, just a couple of brief questions for our evaluation purposes. So I'd appreciate it if you could take a moment to fill that out. And while you do that, on the screen, I'm going to post some upcoming webinars. We have 2 coming up in February, and actually more kind of in the wings. So we will have some coming up in March and April. We like to do our webinars in the winter when folks aren't out in the field. So we have one on pest management strategic plan and crop profiles coming up, and IPM weather map apps is also coming up. Oh, could I put the links in the chat? Yes, I can. And while folks are filling that out, I actually have a question from Lillian for Carlos Quesada. If you're here, just to confirm, she wants to know these parasitic wasps parasite some of the pests of the crops that you studied, correct?
Yes, most all the parasitoid wasps that I use in my statistic analysis were parasitoid wasps that attack aphids.
Okay, great, thank you. Okay, and I'm going to end the poll and share the results. And for anyone who is interested, it looks like most people agreed or strongly agreed that it was increased your awareness of current research and extension, and how likely you to increase your collaboration. We've got a mixed bag on that one. And how likely you to increase your implementation of IPM. That is also a mixed bag. So thank you very much for sharing your thoughts on that. There will be a copy of today's webinar up on our website. It usually takes about a week for us to make the edits and to get that up on our website. Anybody who is registered will receive an email from me with a copy to the link. And you can also go back and see in that same place previous research update conferences. It's always very interesting to me what a mix of research we get on these little short snippets. And so feel free to go back and take a look at other presentations that have happened in other years. So I would like to thank USDA NIFR through the CPPM program for funding for the Northeast IPM Center. And this presentation wouldn't have been possible without their support. So with that, it is 12:02. Thank you very much, and appreciate your attendance, and apologies for getting tongue-tied around a few different words this morning. It was there was a little challenge there. And so I hope that you have a wonderful rest of your day, and thank you very much. Okay, bye-bye.